And if you break the disulfide linkages in the protein, it will make the protein linear. So consider this idea. Let's say this is a protein. This is a disulfide linkage, another unit, right? Disulfide linkage. So BME will cleave it and will separate them as separate entities. So any further structures, further subunits of the protein connected by disulfide bridge will be completely separated as single straight chain of polypeptides. And this is the very first step. We make them linear. The second step, we make them negative charge with the help of sodium dodecyl sulfate, SDS. And sodium dodecyl sulfate interacts to the amino acids. How they interact? Every 2 amino acid, 1 sodium dodecyl sulfate. This is how they interact. So sodium dodecyl sulfate interacts to 2 amino acids. 1 sodium dodecyl sulfate, 2 amino acids, and it carries 1 net negative charge. So the linear proteins that we have, linear polypeptides that we have, they will be filled with negative charges. So whatever proteins we get with the same idea, it will have negative charge. So at this point, if we treat the proteins with beta mercaptolithanol first and then sodium dodecyl sulfate, then we can get rid of the charge density biasness. So it does not matter what is the charge density of the protein. It does not matter whether the protein is globular or fibrous, because they all will be treated the same and they all will be carrying a negative charge. Okay? And generally, if a protein don't have a very high or very low charge natural very low or very high positive or negative charge, then treatment with SDS will make all the proteins almost at a similar charge.